Welcome, everybody. I am so grateful to have been invited once again by Bernie Siegel to present at the World Stem Cell Summit. And the presentation I'm going to give today is a little different from what you're used to hearing me talk about. It's based on my actual research career as opposed to my role as an editor. Most of you will know me as the editor-in-chief of Stem Cells and Development, the Mary Ann Liebert Publishing House title. You might also know me as the executive editor of Nucleic Acid Therapeutics. But today I'm talking to you as a faculty member of the Department of Pediatrics at Wayne State University School of Medicine in Midtown Detroit. What is it that I'm going to be talking to you about? I'm going to be talking to you about the international effort that was put together to try and decode the human genome that was begun in 1990. It took 13 years and an estimated $2.7 billion to complete the gene mapping and the associated DNA sequencing. This achievement was anticipated to catapult our understanding of longevity, disease vulnerability, and lead to precision preventative medicine. But although a stunning achievement, it hasn't delivered as quickly as we anticipated the outcomes that we thought. To quote the great Francis Collins paraphrase, genetics loads the gun and the environment pulls the trigger. Understanding the etiology of disease triggered by exposures to chemical and non-chemical stresses requires a multidisciplinary approach. Researchers in the fields of epidemiology, genetics, epigenetics, genomics, exposure science, toxicology, biology, biomedical science, bioinformatics, chemistry, physics, psychiatry, and environmental justice are all needed to understand how our lived experience, our cumulative lived experience, interacts with our genome to truly understand longevity, susceptibility to disease, and perhaps more importantly than longevity, our well-span. How long can we live as healthy individuals? I am honored to serve as the primary investigator of the Integrative Health Science Facility Core for the National Institute of Environmental Health Sciences Core Center for Urban Responses to Environmental Stresses, the Cures P30 Center here in Midtown Detroit. This role has afforded me 8 years of experience facilitating multidisciplinary, multi-institutional research and team building in response to environmental health concerns. Our successful science, but our continued inability to effectively mitigate the unjust burden of environmental stresses on the people of Detroit, inspired me to alert Mary Ann Liebert, the fierce advocate for impactful health science and owner of the publishing house that bears her name, to the desperate need for a new journal that will provide a much needed home for global research scattered across domains and health disciplines to address the sources, mechanisms, outcomes, and risk mitigation associated with our lived experience. Christopher Wilde in 2005 coined the phrase exposomics to complement the term genomics as a field of research that some have almost defined by exclusion as whatever is not the genome. But we know we can't simply chop one out from the other. What we're looking at, what we're studying, what we need to study in order to understand health outcomes is the interaction of genes and environment. That is why I have proposed, and Mary Ann Liebert has supported, a new title called Gene X Environment, Gene by Environment. For those of you that are longtime attendees at the World Stem Cell Summit, you know that I usually appear on a panel with my fellow conspirators, Alan Giacomo, Jean Loring, and Beth Roxland, and we usually tackle some pretty, well, sometimes some ghastly issues, but all with the view to have a shared discussion from very different viewpoints. Jean Loring, of course, is a very famous stem cell researcher with an interest in reviving rhinoceri, as well as performing wonderful experiments involving space. And Alan is an international lawyer of world renown and interest in everything from AI to, well, now he's going to be very, very interested in gene by environment interactions. And Beth Roxon, a very famous lawyer with specialties in bioethics, very capable. And you should, if you get the chance, enjoy our discussion entitled "Blasting into Space to Maintain the Human Race," and I think you'd learn a lot from it. But I'm gonna recapitulate some of that information here because it truly has a very, very important role to play in understanding how complex it is to understand gene by environment interactions. If you're out in space beyond the magnetosphere, exposure to either microgravity or radiation can result in damaging effects at the molecular and cellular level, changes to cellular structure, differential potential, epigenetic effects, DNA damage response, and oxidative stress. What's more complicated still is that differing stem cell populations in the same animals have differing susceptibilities to those stresses, gravity, and different types of radiation. Different animals have differing abilities to handle radiation exposure. For example, cow oocytes do much better than mouse oocytes in response to radiation. But of course, we haven't even begun to look how human oocytes and embryos will respond. And that's even before we even begin to consider whether individual human populations of differing genetic predispositions would favor or disfavor inclusion in an effort to populate off-world. These are very difficult topics. These are very hard ethical questions to answer, but our ability to understand our genome and how it interacts with our environment will crucially inform those decisions. And back to Earth with a bump, we're going to talk about obesity, a much, much misunderstood condition. I don't think with all the information that's thrown at us on a daily basis concerning food intake, we really take the pause to understand that humanity's natural instinct is to consume food when it is available. There is nothing natural about denying oneself food when one has access. But I'm going to talk about an example that basically centers around people that did not have enough access to food. And this was the Dutch famine of 1944 to 45. Men that were conceived during that famine had higher rates of obesity at 19 years old than those conceived before or after it. So by being in utero of a mother who was exposed to the famine, they then had more inclination to gain fat, which sounds like an entirely understandable response. More interestingly, maternal malnutrition during early gestation was associated with higher BMI and waist circumference in 50-year-old women. So this is a lifelong change in how they've responded. However, men and women born in Amsterdam during the same time, the famine exposure itself did not affect their then subsequent children's birth weight. So those that were in utero during the famine that then had children, their children did not show a difference in the birth weight. But those children of those exposed to famine did increase neonatal fat tissue and were twice as likely to experience poor health in later life than the next generation of women who did not experience famine. That's quite a complicated story, but basically what we're talking about here is that an exposure to an environmental stressor has produced a transgenerational effect. Now, more closer to home, we were all horrified when my former colleague at the Children's Hospital of Michigan, Dr. Mona Hanna-Attisha, and colleagues alerted us that there were elevated blood levels in children associated with the Flint drinking water crisis. One of my fellow Cures researchers, Douglas Rudin, published a paper in Scientific Reports entitled Multi-Generational Epigenetic Inheritance in Humans. And what they showed that the DNA methylations was changed associated with maternal exposure to lead. Not only that, that was transmitted to their grandchildren. Tracy Baker, current Cures member, has also showed that exposures to dioxin can produce multi and transgenerational changes in the methylome. That's basically the characterization of what genes are turned on or turned off in zebrafish that are exposed to water containing dioxin. But of course, we can't talk about Detroit without talking about the effects of proximity to environmental stresses. But it becomes even more complicated. We have to recognize and understand that people of different backgrounds can show differing susceptibilities to develop, for example, breast cancer. Socioeconomic status alone does not predict health outcomes. We have to know the genome of the individuals living in the same neighborhood to understand why certain people develop a health outcome and other people do not. Now, I've mentioned feeding and obesity. I want to now talk about the gut microbiome. I mean, the microbiome, you've probably heard the phrase, it's the sum of the microbes, including the bacteria, the fungi, viruses that live on and inside us, helpful or otherwise. They're there. They may help defend us against pathogens, and without them, we wouldn't be able to digest our food. We are, in fact, more them than us. Humans have, the estimates vary wildly, but there are more bacterial cells in our body than there are human cells. Fun fact for a dinner party. A single course of antibiotics can alter the gut microbiome in healthy people. And it can take months or even years to recover the original species composition. Regrettably, antibiotics, as well as other medications, are measurable in the waters of Detroit, offering the ghastly possibility that an individual who's allergic to an antibiotic might well accidentally ingest it, other than by receiving the medication. The gut bacteria that have antibiotic-resistant genes may pose several threats. When gut bacteria and pathogens mingle, the resistance genes can be transferred, allowing pathogens to acquire resistance. One recent published study observed that resistant gut can even degrade antibiotics in the gut, shielding pathogens from the effects of the drugs. So you see, gene by environment interactions need to take account of not only the environmental effects on our genes, but also our supernumerary multitudinal microbiome and their diverse associated genes. I want to say a couple more remarks about precision medicine and some just fascinating work that Jürgen Hahn is doing at the Rensselaer Polytechnic Institute. Hahn and his colleagues can now predict with a 90% accuracy whether a pregnant mother who has had a previous child with autism spectrum disorder has a 1.7% or a 10-fold increased risk of having a child diagnosed with autism spectrum disorder based on the metabolites of the folate-dependent transmethylation and transsulfuration biochemical pathways. This is fantastic and is getting us further along on the path of understanding who is at risk, who is less at risk. One thing I want to talk about, and I don't think it's talked about enough, is chronology. Knowing when to intervene may be just as important of knowing what the optimal intervention might be for a condition. Classic example of this is a condition like, for example, spinal muscular atrophy, where you know there is a genetic predisposition for a child to develop problems that will include mobility and include the ability for the muscles to even allow the child to breathe. Very, very, very tough decisions have to be made as to, in the most extreme form, whether that child is going to require intubation in order to breathe properly. That decision to make that intubation is not a reversible procedure. So anything that we can understand better about whether a child that has a genetic predisposition to develop spinal muscular atrophy, but that we don't yet know what extreme form the condition is going to take, is going to allow us to make better decisions for the health care of that child. So what I've tried to give you here is a flavor for some of the research that's already going on to try and answer these gene by environment questions. It's not as if people aren't already exploring these questions. They are. They just don't have a journal to publish that work in that is specifically going to focus on that subject. The kind of work we're talking about are genetic determinants of environmental risk, environmental epigenetics, exposomics, chemical and non-chemical stresses and developmental health outcomes, molecular epidemiology, social determinants of health, genome-wide association studies, metabolomics and metabonomics. And very interestingly, what we're going to have to develop are screening tools. We had a glimpse of Jürgen Hahn's work with trying to develop a tool that would allow to work as a biomarker for autism spectrum disorder. But we need other tools as well that are going to allow us to estimate what the level of interaction is. This is also going to take artificial intelligence. It's going to take bioinformatics. It's going to take computer modeling. We need to model what these interactions are to better predict those chronological needs that I was describing when talking about spinal muscular atrophy. We need a truly multidisciplinary international editorial board. We need environmental toxicologists, geneticists, yes, but we also need mathematicians, computer modeling experts, psychiatrists, epidemiologists, pediatricians, chemists, geologists, and environmental justice experts. The list is practically an entire university. To quote with a paraphrase, just as I opened from Francis Collins, I'm going to paraphrase John F. Kennedy. We choose to go to study gene by environment interactions, not because they are easy, but because they are hard, because that goal will serve to organize and measure the best of our energies and skills, because that challenge is one that we are willing to accept, one we are unwilling to postpone, and one which we intend to win. If the aims that I have described resonate with your research goals and you want to be part of this vital intersectional research endeavor, please email me, Graham Parker, at gparker@med.wayne.edu. And let's get to work, advancing our knowledge for ourselves, our children and grandchildren, to live perhaps longer, but more crucially, healthier lifespans, or indeed well spans. What you see here is the research building, the Integrative Bioscience Center that it's my privilege to work at in Midtown Detroit. I just want to say a big thank you to all my colleagues at the Cures program at the P30 Center, our funders from the NIEHS, and for all of you for your kind attention. Thank you very much.